Alright, so the second part of chapter 16 has to do with the structure of DNA and then the replication of DNA. So, Watson and Crick ultimately get the Nobel Prize for developing the double helix model of DNA, and you want to really understand that double helix model. So we have sugar phosphate backbone, then there are nitrogenous bases, hydrogen bonds are what hold those bases together, which is what holds the strands together. DNA coils up to protect those hydrogen bonds. Since hydrogen bonds are weak bonds, they're easily disrupted. All kinds of stuff can disrupt them, including water, which would be bad. And so we coil up DNA super, super tight to protect all those hydrogen bonds and make sure that we only have A's pairing with C's and C's pairing with G's. But now, how do we get more DNA? That's the next question, because if DNA is, in fact, our transforming factor, our genetic information, our inherited factor, then we have to get more of it in every single one of our cells. So, we have DNA replication. And there are 5 basic steps. First thing, we have enzymes that get us to untwist the helix. Then we separate the helixes, or the sides of the helix. Then we add the nucleotides. Then we proofread it. And then we twist everything back up. So that's the basics. Now, this is exactly it. So we have our strand of DNA, separates, nucleotides come in, they're complementary, and we're done. But there's a little bit more to know than that. And so we went over this in class, but you want to make sure you can hear it again and practice and know it all. So the little green enzyme, it doesn't have a label. I don't know why. It's helicase. It's the one that starts the whole thing, so it's responsible for that untwisting. Then we have single strand binding proteins, those little black, sort of black-gray things there, and those are proteins that hold it open. And then we have 2 strands of DNA. 2 different things are going on here. First, we have the leading strand, and then we have the lagging strand. And so for our leading strand, think of it like on a train track. And so since DNA polymerase III is going to just add nucleotides in a complementary strand in the direction of 5 to 3, so it's adding nucleotides 5 to 3. It's reading DNA 3 to 5, really important. Reading DNA 3 to 5. Adding nucleotides 5 to 3, then on the leading strand, it's just going to chug along. So it's the direction that DNA is separating. It's just going to keep moving in one single piece. Now, how did DNA polymerase get started? Well, DNA polymerase, being an enzyme, simple machine, can only do one thing. It can only add nucleotides to an existing strand. So how do we get the first nucleotides if it can only add to the existing strand? That's where our pink enzyme comes in. Primase. So primase puts down an RNA primer. Why RNA? Because, who knows? And so with primase, it can only add RNA. It only adds a couple of nucleotides of RNA. It can start with nothing there before, puts a couple of RNA nucleotides on, falls off. So it does that even on the leading strand, but it only does at the very beginning of the leading strand that's covered up by the yellow picture. Then, after those few RNA primers are there, since DNA polymerase III only adds nucleotides to existing nucleotides, it can come in and start adding DNA nucleotides. So it keeps doing that, keeps doing that, keeps doing that, until it either runs out of space or until it runs into other nucleotides. And so on our leading strand, it's just going, 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 going. On the bottom, we have our lagging strand. Why is it called the lagging strand? Remember that DNA reads in the 3 to 5 direction, and it adds nucleotides in the 5 to 3 direction. And so when we look at our strand, to read it, to go along in our 3 to 5 direction, then we have to be going backwards. And so we have to be going along and sort of working in opposite direction. And so we're reading DNA 3 to 5. Adding nucleotides 5 to 3, which means we can only work little segments at a time since it's an anti-parallel molecule. And so then we have all these little pieces. So we have primer, added by primase, DNA polymerase III coming in, adding those DNA nucleotides until it runs into another nucleotide, whatever it is. Seems fine. Now, how do we get rid of that RNA? How do we get rid of those red primers? We have another simple machine. So DNA polymerase I is going to come in and replace those RNA nucleotides with DNA nucleotides. Why doesn't DNA polymerase III do that all the time? Or why doesn't DNA polymerase III do that It's a simple machine, it's not its job. Why doesn't DNA polymerase 1 just add all of the nucleotides? It's not its job. It can only take out one nucleotide and put in the DNA replacement for that nucleotide. So think super simple machines, they can only do one job. So now we have all of this DNA, but on the lagging side especially, we have all these little pieces. That's where Okazaki fragments come in. They're just little pieces. And so we have all these little pieces of DNA. Well, how do we link all the pieces into one giant strand? Another enzyme, DNA ligase. So ligase comes in, adds that one or adds those backbones, links them together, but only after they're all DNA. And so then we have two complementary strands of DNA. So big molecules to know. Helicase is the purple one. Helix, so it has something to do with either twisting up or untwisting. That's what it does. Single strand binding proteins hold everything open. DNA polymerase 3 adds complementary DNA nucleotides. Primase adds RNA primers to get started. We need RNA primers for DNA polymerase 3 to bind to. DNA polymerase 1 is going to replace RNA nucleotides with DNA nucleotides. And DNA ligase is going to link those backbones together. We didn't talk about DNA polymerase 2. What the heck? DNA polymerase 2 is a proofreader. And so its job is to come in and proofread to make sure that we didn't make any mistakes. Why is it so important? DNA is your genetic information, and so we need it to be as error-free as possible. Do mistakes still happen? Absolutely. You can pay somebody $1 million to be an editor, and they're still going to make a mistake every once in a while. Just hopefully if you're paying them $1 million, they're not going to make many mistakes. Your simple machine, its only job in its entire existence is to proofread. So most of the time it gets it right, but every once in a while it makes a mistake. If it does, comes in, excises, takes out that bad nucleotide, fixes it, then you should be back to normal. Ligase, by the way, is going to come in and link those pieces back together. So, should be good.